So yeah, get that in if you haven't yet. I think that's it. Those are the three main announcements I had. Any questions before I go into the cell structure? I don't think we started on this at all. I'm a little bit behind in this class compared to my 9 a.m. I think that's what I seem to remember. But we should get through it. Should get through it today. I did change, if you downloaded this PowerPoint like a while ago, I've had it up there for a while, but I changed in the past couple days. I just cut some stuff out and simplified it. So if you see some stuff, if you have it printed out or an older version downloaded, you might see some stuff that I don't have in here. This correct updated one is now on Canvas. So anything that you see that's not on here, you don't need to know. All right, so let's see. We were talking about chemistry. Now we're moving up to the cell level. So entire cells. And we're going to start out talking about kind of the different parts of the cell membrane and then how stuff gets transported across it. First off, a few concepts you need to know about cells are based on the cell theory. So you may have heard this before. There's three main principles that in biology, we think of these as true about cells. So this is a theory of what cells are, kind of how they work. All organisms are composed of cells, so at least one cell. Bacteria are single-celled organisms, so they're only made of one cell, but they're still alive. That makes the cell the simplest unit of life. And then the only way to get more cells is to have a cell to begin with. So cells make more cells. You don't just have a cell coming out of nowhere. So for our purposes in today's day and time, cells make more cells. There are many, many different types of cells. We're going to sort of talk about cells generally in this chapter, but we'll get into tissues after you take the exam on Monday. Our first chapter after that, I think, is tissues, and we're talking about it in lab this week. There's all kinds of different cells in your body that do different things. So it's not like one cell, there's only one kind of cell. There's 200 types of cells. You can have nerve cells, you can have skin cells, heart cells, blood cells. They're all slightly different and have their own functions based on their specific structures. So there's a bunch of different cell types. Right now, we're just keeping it very general. The main components of the cell, if we break it down very broadly, the plasma membrane or the cell membrane is the border around the cell. We're going to go into some detail on that today. It's primarily made of lipids. It also has proteins in it, but proteins and lipids make up the cell membrane. The cytoplasm is sort of everything within the cell. That's where the organelles are found. So like the nucleus, the mitochondria, all of those that we'll also cover. Those are in the cytoplasm. I won't really use this term. I have it up here in case you see it, cytosol. That's the fluid part of it, like a gel-like substance. I'll pretty much say intracellular fluid or ICF. You'll see this abbreviation going forward. ICF is the fluid inside the cell. We'll contrast that oftentimes with the fluid outside the cell, which is extracellular fluid. So intra, inside, extra, outside. ICF and ECF, you'll see those abbreviations going forward. So extracellular fluid is just the fluid surrounding the cells. Differences in concentrations of molecules and solutes inside and outside the cell will allow for basically movement across the cell. So we'll look at those in relation to each other kind of throughout the rest of the semester. We're going to start with the plasma membrane. Most of the molecules in the plasma membrane are those phospholipids that we talked about on, what is it, on Monday. There are these really interesting, unique lipids that have a head and then two tails. So if you counted up all of the molecules in the membrane, 98% of them are these phospholipids. They're pretty small, but they make up most of the volume. Sorry, 75% of the lipids. So 98% are lipids, 75% are phospholipids specifically. There's another kind of lipid I'll talk about on the next slide. But the vast majority of a plasma membrane is phospholipids. A couple of important aspects of them: they have a hydrophilic head, hydrophobic tails. That makes them what we call amphipathic. This term, amphi meaning both ways, so kind of two paths, the hydrophilic side and the hydrophobic side. And they're arranged in a bilayer. So it's not just one layer of these guys, there's two of them, as you can see here. So if this is a plasma membrane, this would be outside the cell, this would be inside the cell down here. The hydrophilic heads are sticking out towards the fluid, which is mostly water. The hydrophobic tails are scared of water, so they're facing each other. They're not interacting with the water. So tails are facing inward, heads are outward. Yeah. That's the basic setup of most of the plasma membrane. Other lipids in the membrane include cholesterol and something called glycolipids. I mentioned cholesterol on Monday as well. This makes up about 20% of the lipids in the membrane. So there's a good bit of cholesterol in the plasma membrane, and you can see them in this image. They're these little blue rings, basically, the stack of rings. Those are cholesterol molecules, just interspersed in between the phospholipids. So all of these yellow things are the phospholipids. Then you have some cholesterols just stuck in there in the middle. They're going to help to keep those phospholipids in place and kind of solidify the membrane just a little bit. Not too much, but just a little bit. And then finally, we have some lipids that have carbohydrates attached. That's what these green sticks sticking off the top are. That's a carbohydrate chain attached to a lipid, a phospholipid. Those are glycolipids. Anytime you see glyco, think like glycogen, carbohydrate, it's a carbohydrate. So glycolipid is a combination of this carbohydrate attached to one of the phospholipids. They're important in cells communicating with each other, essentially. So communication functions. They're like little, I don't know, antennae, I guess you can think of them, antennas sticking off the membrane of the cell, kind of sending messages out. Those are the three lipid types. That's all I'm going to say about the lipids. Any questions about those? Okay. Next, we have the proteins. So the proteins are very important. Not that the lipids aren't. They make up most of it, so I don't want to brush by them too quickly. And next, we have membrane proteins. They only make up 2% of the total number of molecules. So there aren't many of them, but they're gigantic. They make up 50% of the weight of the membrane. These colorful things here are just drawings of different kinds of proteins. You don't have to know all of these different types of proteins that are listed along the bottom, kind of just there for your information. We're going to go into some basic functions of these proteins over the next few slides. So not many proteins, but they're really important. They allow for passage of material that can't go through the membrane on its own. So there's some molecules that can go through the phospholipid bilayer, but a lot of molecules are either too big or will get bounced off that hydrophobic tail region. So they can't go through on their own. These proteins are like little helpers. They're little channels that allow for a lot of molecules to go into or out of the cell. That's the purpose primarily of these proteins. And that's where we get into membrane transport. Moving things across the cell membranes is critical. You need to get waste out, you need to get nutrients in, you need to keep the solutes balanced on both sides, et cetera. So there's a lot of movement across cellular membranes. We call the plasma membrane selectively permeable. That just means permeable means stuff can pass through it. Selective, obviously, it just doesn't allow everything through. It allows very specific things through and keeps others out. I'm going to talk about a couple of general categories of membrane transport, passive and active. Those might sound familiar to you. Anything that is transported passively doesn't require ATP, so no energy. It just happens without energy input. Active mechanisms do require some kind of energy input. There's three subcategories of passive mechanisms I'm going to talk about: filtration, diffusion, osmosis. Active mechanisms, I'm just kind of all lumping together. We're just going to talk about active mechanisms as kind of one big category. A special subset that can include both of these would be carrier-mediated mechanisms. And that just means anything that's carrier-mediated, it means a protein is needed. The protein is the carrier. It's mediating the transport. It's allowing for transport of something across the membrane. It can be active or it can be passive. So the protein might need energy to do that, or it might not need energy to do that. It can be either. So carrier mediated can fall under either of these, passive or active. The main thing is you need a protein. That's the idea behind carrier mediated. So I'm going to start with the passive mechanisms. Go through each of these, kind of describe what they are, how they work. First, we have filtration, which you may or may not have heard of. I don't cover it, I don't think, in general biology, but it's a really simple concept. It's just pressure pushing things out of cells or through cells, basically. So if you think of like a blood vessel, there's a lot of pressure running through your blood vessels. That pressure will push water and other types of solutes out of the blood vessel, either through kind of cracks in between the cells, cell junctions, or through the cells themselves that are creating and lining the blood vessel. So it's just physical pressure pushing things out. That's filtration. They have to be pretty small particles, obviously, to make it out. Anything big like a blood vessel is not going to get squeezed out of those tiny pores. And that's all I'm going to say about filtration. It's all just driven by physical pressure. Diffusion and osmosis are both dependent on solute concentrations, so I'm going to spend a little bit more time on these. Simple diffusion is where you have movement of a solute particle, we'd say down its concentration gradient. So from a high concentration of itself, like on this side, if this pink thing is a membrane, these are solute particles, these little blue donuts. Over time, simple diffusion will predict that these particles will move towards where there's fewer of them. So they'll go from a high concentration of themselves to a lower concentration of themselves, and then it'll eventually even out. So both sides of the membrane have the same solute concentration. So we say that's movement down the concentration gradient, or you can say with the concentration gradient. You might see that term. This happens naturally. So this is what particles do on their own without needing any energy. So this is why it's passive. Particles naturally want to spread out from themselves. That's simple diffusion, pretty simple. Osmosis is really closely related, but here we're talking about water. So when you see the word osmosis, think it's the movement of water somewhere, and I'll talk about where. So it's the movement of water through we'll say a cell membrane, a selectively permeable membrane. And what water does, so if solute particles move in simple diffusion from an area where there's a lot of them to where there's not a lot of them, water will do the same thing. If you think of water as kind of individual particles, like these little blue dots are representing water here. So a water molecule, wherever there's fewer water molecules, that's where the water will move in that direction. It's kind of weird to think of it that way. But if you have fewer water molecules over here that are free, and you have a lot over here, over time, you're going to get movement to the right across this membrane, assuming this dotted line is a membrane, and assuming the solutes can't move back and forth. So these big orange or reddish color circles, those are solutes. They can't go across the membrane, only water can. So you'll get a rise in the level of water on the right side here in this case. So water moves from where there's a lot of it to where there's less of it. A more, I think an easier way to think about it is that water is going to move towards where there's more solute. And when we talk about hypertonic and hypotonic solutions, I think on the next couple slides, that's, I think, the easiest way to think of it. Wherever there's more solute, that's where water is going to move. It wants to dissolve the solute. So in this case, there's a lot of solute over here, so water is going to move in that direction. And this is really critical for creating and producing IV fluids. So this goes back to that video I showed you guys, I think on the second day, about Emily Jerry and the IV solution that was put into her veins was like super, I think, hypersaline, he said. So there's far too much salt in it. And that basically drew the water out of her body cells and led to her death. Osmotic imbalances aren't necessarily always that dangerous, but they do underlie a lot of issues that you can have with your body. So constipation, diarrhea, those are kind of on opposite ends of the spectrum. And then edema, which is swelling. You want to know that term, that'll come up again, edema. And that's just where a lot of fluid is collecting in one area of the body due to potentially a lot of solute in that area. All right, so that's the basics of osmosis. Now we're going to talk a little bit more specifically about this idea of hyper, hypo, and isotonic solutions. So this is where you can predict which way the water is going to move. And in our case, we're kind of thinking about body cells in some kind of solution. So our body cells are surrounded by fluid, by extracellular fluid. There's solute inside our cells and there's solute outside our cells. Extracellular fluid and intracellular fluid. Both of those have solute particles. Depending on which side of the cell membrane has more solute is going to determine whether water is going to move out of the cell or into the cell. So if we think of, we'll imagine one individual red blood cell, we stick it in a solution, a beaker, and that solution is hypotonic. So a hypotonic solution. Hypo means low, tonic refers to the solute concentration. So low solute in the solution surrounding the cell. That is going to mean there's more solute in the cell. So more solute particles are in the cell. And water is going to flow towards that higher concentration of solute. So it's going to go into the cell. So this is always relative, right? If you have a hypotonic side, you have a hypertonic side on the other side. So we could say hypotonic solution around the cell, and then the cell itself, the intracellular fluid, we could say is hypertonic. It has more solute in it than outside of the cell. So you always want to think about it in relation to kind of the other side of the membrane. So hypotonic solution means the extracellular fluid is low in solute concentration. The opposite would be the case if you have a hypertonic solution. Hyper means a lot. So if we look over here, we have a whole lot of solute particles outside this red blood cell. That means water is going to flow out of the cell towards that high solute concentration. So you get a lot of water leaving the cell, and that's going to shrivel. So it's going to make the cell shrivel down. Isotonic is really easy. Iso means the same. So solute concentrations inside and outside the cell are the same. Water's moving, but it moves equally. It's not like more moves out or in. It's an equal exchange of water. And that is what most of our blood cells are happiest in. So red blood cells want to be in an isotonic solution. Most of our body cells in general want to have approximately the same concentration of solute inside of them as outside of them. So normal saline is what goes into IVs. That's 0.9% NaCl. And that's about what the concentration of NaCl is inside of our cells. So by adding a very specific percentage of solute to our bodies, we can hydrate body tissues without throwing off the osmotic balance. Okay, that's really all you need to know about hypotonic, hypertonic, isotonic. Any questions about those? Good. Okay, into actually using protein. So all of that I talked about just then was kind of movement across a membrane, but we didn't even mention proteins, right? No proteins were involved. Stuff was just moving across the membrane, solutes or water, without the help of proteins. Now I'm going to talk about the use of proteins in carrier-mediated transport. So there are these helpers that allow things that wouldn't normally be able to cross the membrane to cross the membrane. So I think of them as like little tunnels that allow for the passage of materials that normally wouldn't make it through the phospholipid bilayer. Oh yeah, I have 3 here. I should change that. Sorry, I got rid of 1. I'm just lumping all of active transport together. So we only have 2 mechanisms of carrier-mediated transport. I changed that pretty recently. Forgot to change that number. So 2 mechanisms of carrier-mediated transport, basically passive and active. So the passive is just diffusion, same idea. Something, solute is moving from a high concentration of itself to low, but it has to move through a protein to get across the membrane. So simple passive diffusion through a protein is called facilitated diffusion. The protein is facilitating the movement, it's helping it. No energy required. It just does it naturally. It just needs that little tunnel through the plasma membrane. I don't think I'm going to talk any more about facilitated diffusion. Just know that it involves a protein and it's passive. Active transport also involves a protein. So any kind of active transport, I'm only really kind of talking about one example, but anytime you have active transport, you need some kind of protein to make that happen. And then that is also going to require ATP. So diffusion can happen with or without a protein. Active transport, you have to have a protein. So with active transport, now the carrier, the protein, is moving some molecule up or against its concentration gradient. So I mentioned that naturally molecules want to kind of spread out, right? That's diffusion. That's what happens naturally. If you want to do something that's not natural and move a molecule to where there's a greater concentration of it already, that's where it takes energy. It's going to take ATP to make that happen. And that is moving it up its concentration gradient. The example I'm going to mention, and you've probably heard of before, is the sodium potassium pump. Does that sound familiar to anybody? Sodium potassium, awesome. You probably learned about it in a general biology class. And it's a great example of active transport. So the idea of the sodium potassium pump is to get as much sodium outside the cell and as much potassium inside the cell, not as possible, but to create, well, I'll talk about in a second. So the cell wants to get rid of sodium and bring in potassium. And both of those are going against their concentration gradient. So we'll go to the next slide before I go into this. So sodium potassium pump, you're moving sodium out. You're moving potassium in. Moving sodium out, there's already a lot of sodium out here. So naturally, sodium would kind of fall back into the cell if we're following simple diffusion. So we're going to use energy, ATP, to kick even more sodium out of the cell. And same for potassium, using ATP to bring more potassium into the cell. So you get a high concentration of potassium inside the cell and a high concentration of sodium outside the cell. Creating this, like setting this up, is what allows our nerves to fire. It's what allows our muscles to contract. So it's critical throughout our body to allow us to function. What it's doing is, the idea is to maintain a negative charge inside our body cells. So here you can see 3 sodium. Sodium and potassium are both positively charged ions to start with. So they're both positive. You kick 3 positives out, 3 positive sodiums out. You only allow 2 potassiums in. So 2 positives come in. That's going to bring the charge inside the cell down, down, down with each cycle of this happening. And it happens all over the cell millions of times a day. So more positives are going out than coming in. So you get a negative charge inside the cell. We call that a negative resting membrane potential. We'll talk all about resting membrane potentials when we get into the physiology of muscles and nerves. But really all you need to understand now is that it creates this negative charge inside of a cell. Outside the cell is positively charged. It sets up a scenario kind of like a battery. You have a negative and a positive side. And that's what allows for eventual contraction of muscles or firing of nerves. So think of it as a battery. And this is a pretty fascinating fact. About half the calories you take in every day go towards setting up this negative membrane potential, to kicking 3 sodiums out and bringing 2 potassiums in. So half of what you eat is going towards your sodium potassium pumps in all your cells. That's pretty crazy. Any questions about that? That's really all I want you to know for now about the sodium potassium pump. It'll come back up, so don't forget about it. I'll remind you of it when it comes back up. But those are the basics for what you need to understand right now. Okay. The cell's interior. Everyone has learned the organelles probably in high school, probably in college as well. So instead of me going through these organelles, we're going to play bingo instead. It's going to be at least slightly more fun than me talking about all of the organelles ad nauseum. So I'm going to hand out the sheet that has words on the front, and then you're going to make your own bingo board. So you're just going to put all of these words into one of these squares. So there's 16 words, there's 16 squares. So fill it in however you want. There's no rhyme or reason to how I ordered these, really. all of those words should fit on that board. And don't forget the free spot. You get one free one. So these are all organelles or parts of organelles. You can use your notes for this. I've printed out the slides from the rest of this presentation, which has all of the information you need for these organelles. You don't need to know anything additional. Does anyone want a copy of these? Okay. For the game, you can use your notes. You can ask your friends. So all the clues that I wrote are from these slides. Starburst for whoever gets the first few bingos. Well, everyone will eventually get Starburst, but. You get to pick out your favorite flavors before they are all gone. So you get one of the first bingos. So I have 15 clues up here. We won't get through all of them. We don't quite have that time, but I'll post this to Canvas and the answers are in the little note section under each slide. So you can kind of quiz yourself. But each of these clues is sort of a description of an organelle that you have on your board or a part of an organelle. They're not all specifically organelles. There's a few that aren't. So as we go through these, just cross off if you have, like, you know, bingo, if you haven't played bingo before, and cross off the square that describes or that is the name for what I'm describing in these clues. And it might be helpful to write the number like in the corner of the square, the clue number, just so we'll go back once we get in like 10 minutes, we'll go through the answers to the ones we got through. So you can kind of go check and make sure you linked up the correct clue with the correct name. If you get bingo, obviously yell bingo or organelle or whatever you want to yell. I think that's it. Is everyone about ready? Okay. And all of this information, like I said, you can use help from your friends, you can use your notes, whatever. This is sort of how we're going over the organelles rather than me talking about them. Here we go. So number one, double membrane that regulates molecular traffic into and out of the nucleus. I'll give you all about 30 seconds for each of these, and then we'll move on to the next one. All right, number two. These are the monomers of a protein. So obviously it's not an organelle, but closely deals with something in the cell. Ready? Next. I don't want to go too fast. Is everyone pretty good? Okay. A membrane-bound organelle that breaks down the cell's waste. So what breaks down cellular waste? Ready? Next. A membrane-bound structure that transports completed proteins to the cell membrane for export out of the cell. That's kind of a tough one. It's not a formal, like a formal organelle, kind of part of an organelle. Next. No bingos yet? That was the first opportunity with 4. Okay. An organelle that directs most of the cell's activities. Next, series of channels attached to the outside of the nuclear envelope with ribosomes attached to the channels. Okay, ready? Number 7. Oh, number 4. You ready? I'm gonna go to 7. 7 is the gel-like substance inside the cell that contains the cell's organelles. We just went over that actually at the beginning of today. No bingos yet? Ready for 8? A membrane-bound organelle that detoxifies substances and is specifically found a lot in the liver and kidneys. All right. Just one? All right, grab some starters. Grab a couple. You can choose your favorite flavor. All right, we have a couple more minutes, so I'm going to keep going. Yay. Okay, can you pass it back that way? Yep, thanks. All right, number 9, the organelle on which amino acids are assembled into proteins. There's potentially a couple answers here. One you may have already crossed off. There's one specific one that's the most obvious.
You said the answers are going to be on it?
Yeah, I'll show you where they are. We're going to go through them in 1 minute. Well, over the ones that we've done already. Nothing? Okay, we'll do one more and then we'll go over these first 10 answers. The organelle that adds finishing touches to proteins. It splices them, it adds carbs, among other things.
Bingo.
Yay. You pass the Starburst up, thanks. Okay, we'll go backwards. What is this one? Anyone know? Well, Golgi apparatus. Yep. Golgi apparatus. Number 9, where are amino acids made into proteins? Ribosome. So the rough ER has ribosomes on it. So I could see how that would be an answer as well. I would accept that, but I was thinking ribosomes here. And rough ER is a little bit earlier. What detoxifies substances specifically in the liver and kidneys? Peroxisomes, yeah. So lysosomes and peroxisomes both kind of do something somewhat similar. Peroxisomes do a lot of detoxification in the liver and the kidneys. What's the gel-like substance in the cell? Cytoplasm, yep. A series of channels with ribosomes attached. Rough ER. So yeah, that was the rough ER answer. And ER is endoplasmic reticulum. There's also smooth ER. They do slightly different things. You want to know those kind of slight differences in their functions. What directs most of the cell's activities? The nucleus, like the autocrat, I guess. The leader of the cell. Membrane-bound structure that transports completed proteins to the cell membrane for export. Golgi vesicle, yeah. So Golgi vesicles will take proteins after they're finished being completed in the Golgi apparatus to the cell. When I was typing in the answer, vesicle, Golgi vesicle, it auto-corrected to Golgi vehicle, and I was like, oh, it's kind of it's a good way to remember it. It's a vehicle to take proteins to the cell membrane.
So the complex is the vesicle?
No, the complex is the apparatus, sorry, yeah. Golgi complex, Golgi apparatus, that's the whole organelle. The vesicle is the little, it like buds off from there.
That's right.
Yeah. Membrane-bound organelle that breaks down the cell's waste. Lysosome, yep. Lysosomes, those are kind of the waste control areas of a cell. The monomers of a protein. Amino acids, yay. A lot of people do that. And then what is around the nucleus? Nuclear envelope, yep, or nuclear membrane, whichever. Yep, awesome. Okay, so I'll upload this and you can see down here. I have the answer to each of them in the notes section, so you can look at the last 5 that we didn't get to, kind of use that as a quiz. If you didn't get Starbursts, feel free to pass them around, grab 1 or 2. And I will see y'all on Friday for review for the exam.